Hello, everyone. I'm Diane Webb, and I'd like to thank you for joining us today for a BizDash webinar. I'm going to start by going through a presentation that I gave last month at the IC-SDV conference in Nice. And after the presentation, I will then hand it over to my colleague, Matt Eberly, who will go through a demonstration of extracting the highest phase for an indication, and then some visualizations that you can do with the BizInt Smart Charts tools once you've done that. So this is the third of the presentations that I gave at the conference in Nice about the challenges of visualizing drug pipelines. And it presents some of our latest developments and the lessons that we've learned over the past 5 years of helping people visualize pharmaceutical information. So in 2013, I talked about how standard visualizations developed for scientific data and even patent data are rarely applicable to drug pipeline data or need significant changes to tell the drug development story. And here's some examples of the types of visualizations that you often see for scientific and patent data. And these tend to be exploratory visualizations showing quantitative overviews of a data set. But here, as many of you know, are examples of drug pipeline visualizations that you see in pharmaceutical annual reports or slide decks. And these generally aren't visualizations that you can build with Spotfire or similar toolkits. They're often drawn by hand in PowerPoint, and they show mostly qualitative or relational information on a few selected items. Now, our goal at BizInt is to reduce tedious manual work like this. So in 2012, we started working with the VantagePoint team to develop tools to facilitate these types of pharmaceutical pipeline visualizations. And in 2016, I presented the second paper at the Nice conference describing our efforts to automate the creation of product launch timelines. And the result of this project was that we were able to automatically draw a very nice timeline in PowerPoint, but a lot of data cleanup was required before you could push the button to generate the timeline. And you can find the slides for the 2013 and 2016 papers, as well as the slides for this presentation on our website at bizint.com/slides. Now, we continue to work on adding visualizations, and in this presentation, I'd like to share our latest developments and lessons learned. So one conclusion of our 2016 project is that many different skill sets are required to create a successful visualization. You need someone who understands how the visualization is going to be used to make decisions. And you need an intelligence analyst who understands the disease area and company interest. A search specialist who knows how to create an accurate and comprehensive search that will give you the data that you need for the visualization. And someone who understands how to use the tools to manipulate the data to create the visualization. And ideally, these skills will be combined in 1 or 2 people, but increasingly, those people are hard to find and they're very busy. So last summer, we created a new service concept, BizInt Smart Strategy Dashboards, and designed for those organizations that are too busy to master all the skills needed to create these visualizations. And the idea is that BizInt will apply our tools to your data to create a suite of visualizations addressing different aspects of a topic of your choice. And here are a few examples of BizDash visualizations that we created for a case study on drugs in development for Merkel cell carcinoma. using clinical trial data. This is what we call a piano chart, showing ongoing trials for Merkel cell carcinoma in each phase, color-coded by trial status. About that. Planned, ongoing, completed, etc. And here is a trial completion timeline showing when trials in each phase are expected to complete, again color-coded by trial status. And here we're using a standard bubble chart to show the companies developing Merkel cell carcinoma drugs and how many drugs they have in each phase. And here we see another type of trial timeline showing trial duration, again color-coded by trial phase. So initially we developed interactive visualizations in Chart Director and JavaScript, but we quickly learned that it's actually all about PowerPoint. People say that they'd like interactive visualizations, but in the end, what they really need are slides for PowerPoint presentations. And you can put a static image in a PowerPoint slide, but it's better if the visualizations can actually be edited in PowerPoint. So our most popular visualization currently is the piano bar chart. It's drawn automatically in PowerPoint, and each drug is a box in a column for that phase, labeled by the drug name and your choice of 1 or 2 additional fields, such as company. And the boxes are color-coded by a field of your choice. In this case, it's type of molecule. And you can have links to the backing pipeline or trial record. But for your final presentation to management, you might want to indicate some additional data, such as who are our key competitors in this pipeline. And by creating this slide as PowerPoint objects, it's easy to manually draw dashed lines around the key competitor boxes. Now, this is something that we could do programmatically, but what if your client wants you to indicate key competitors like this? So we definitely don't want to program 20 different ways of showing key competitors, but it only takes a minute or so to add these types of flourishes manually in PowerPoint. Now, I should note that the databases won't tell you who your key competitors are. It's the analyst's job to know that or figure it out. Now, this visualization looks fairly simple, but unfortunately, we've learned that it's not actually easy. Everyone wants to just type Merkel cell carcinoma into a box, select piano chart, and bang, there it is. But while we've made the drawing part easy, you actually have to do a lot of cleanup to the data before you can draw the final visualization. You need to filter the data to the most critical items, the key drugs. You need to group values in order to create useful categories for things like color coding. And you need to shorten labels, drug names, company names, etc., so that they fit into the boxes. And finally, you often have to apply your expertise to create values which aren't actually in the data. So here's an example of some of the data issues that we had to deal with before creating this migraine launch timeline. The key value for this visualization, which is the estimated launch date, isn't actually a field that the drug pipeline databases provide. And in order to create the lanes, we call them swim lanes, in which the drugs appear, we had to group these target items into a few categories. And likewise, we had to take the route of administration values provided by the databases and group those so that we had a useful number of colors for our color code key. So here's what happens when many of our users create their first piano chart. This is the pipeline for mesothelioma without doing any data cleanup. And VantagePoint and PowerPoint will cheerfully create this visualization, but of course this is not what you want to deliver in your PowerPoint slides. So one problem is that there are 64 drugs color-coded by 58 different technologies, of which you see just the first few here. And what we need to do is group these 58 technologies into just 6 drug types so that we have a meaningful color code key. And this isn't a trivial task. VantagePoint provides some nice tools to do this, but using them requires a knowledgeable and confident analyst. So also pipelines aren't nicely always distributed by phase. And here the visualization is, go back there. Here the visualization's being distorted by having a large number of phase 2 drugs. So we'll split phase 2 into 2 columns, which gives us a more readable and attractive piano chart. Okay, let's talk a little bit more about color. Experts tell us that a color code key should have no more than 8 values, and fewer is better. If your key values have a natural order, such as dates or phase, then you want to use a sequential color key like the ones shown here, so that the eye sees the progression from low to high. But if there's no order to the key values, such as for companies or technologies, then you want to use a qualitative color scheme where all the colors are ideally equal. And these examples come from the Color Brewer website, which is, that's just one of the tools that you can use to construct a color code key. Now, since this pipeline piano chart is color-coded by type of molecule, we've used a qualitative color code key. But this trial piano chart is color-coded by trial status, and that has a natural order from planned to completed. So we've used a sequential color key. And note that we have 2 status items, unknown and terminated, that don't really fit in the sequence, and that we decided to put at each end of the key. And that's just one more example of the sort of decisions that the analyst has to make when preparing the data for the visualization. So what's the best visualization to use? Well, of course, it's the one that is the best fit for your data and the questions you're trying to answer. We like to do some quick data cleanup and start generating different visualizations with different categorizations and color keys to see, what looks interesting. And remember that a visualization that works great for one project might not work well for your next set of data. For example, if all the drugs are in the same phase, the piano chart's not going to look very interesting. So in the 2013 and 2016 papers, I talked about the very popular bullseye pipeline visualization. There's just some examples here. This looks like a polar scatter chart at first glance, but it's actually quite different. The radial values are roughly related to time, and that the rings represent progress from early stages to drug approval. But around the circumference, we have discrete segments, as in a pie graph. And each segment represents a different mechanism of action, target, or even company. And there's often no meaning to the order of the segments or even the size. So it's pretty clear that these bullseye visualizations are usually drawn by hand, often in PowerPoint. Now, for a pipeline with many drugs like mesothelioma, a bullseye might actually be a better fit. And I'm very pleased to announce that we can actually now build a bullseye visualization automatically in PowerPoint as part of our BizInt Smart Strategy dashboards, like the one that you see here for the mesothelioma pipeline. And to say a little bit more about that, BizInt can now build these for you if you're a VPSCE or BizDash customer for the next few months. If you send us your BizInt chart file and your VPSCE file, then we can send you back the PowerPoint slide with the bullseye visualization. And because it's something that we're building in PowerPoint, you can actually fiddle around with moving some of the labels, even doing some things like changing colors and so forth yourself. And then we're working on kind of packaging this up as a script in VPSCE, and we'll deliver that late summer or next fall. So to wrap up, I'd like to thank all the chefs on the BizInt and VantagePoint team who helped develop these visualizations and deliver them, and especially our top chef, Matt Eberly, who created most of the visualizations that you've seen in the presentation and who helps customers create their own. And before I turn the presentation over to Matt, can take a few minutes. If there are any questions, you should see a little panel at the bottom of your screen where you can ask a question. And of course, you can always email us at support@bizint.com. And again, a reminder that the slides for the presentation in Nice and also the 2013 and 2016 slides are up on our website at bizint.com/slides. Okay, I'm not seeing any questions, so I think without further ado, I will hand the presentation over to Matt.
Okay, thank you. Let's see. Hopefully you are seeing my screen. Hello everyone, thanks for coming. My name is Matt Eberly, product specialist with BizInt here, and I want to give you sort of a little behind the scenes, some of how we created some of those visualizations that you just saw, beginning with extracting the highest phase for an indication, which I realized I actually have a updated script there to show as well. That makes things just a little bit faster and very handy thing to do. I think we'll create a small bubble chart comparison, so you can sort of see visually that this has an impact as well. Aren't seeing my screen, you can wave hands or use chat. I think all those options are available to you, but provided things are working as expected, I'm going to shift over and let's take a look. This chart happens to be on mesothelioma. It's a pipeline plus chart, and that means we will get additional matching drugs even if our indication wasn't found. So for example, row 2 here, my search for mesothelioma did find this drug. Pipeline Plus also brought in this additional drug, matching drug record from another database. Okay, so before we go to VPSCE, we want to make sure that we have the columns that we're going to need, because whichever columns are visible here will be available as fields in VantagePoint. So I need drug development phase, and it's nice to have highest phase as well. If I didn't, I could always go into view columns and add them, but it looks like we are ready to go. So I'm going to choose that last export option. You need to save a VPXI file to go in between the two. Name doesn't matter so much, but as with so many other things, I do find myself sometimes going back to re-import a file. So if I give myself a meaningful name, it will be easier to understand if I want to use that again. VantagePoint should launch automatically and import. As you can see, these are the fields that were columns in my chart. And the one thing that I need to do in order to filter drug development phase is I want to clean up this indication here. So I can right-click and choose List Cleanup. General is the fuzzy match file you generally use. And that's going to try to match things up for us automatically, which right now I don't care about because I only need to clean up my target indication. So I'm going to put something in the search box, click the Add button, which is the search button, change to Custom Set of Items, which is the search results, and find that, in fact, this is a relatively straightforward indication. I really only have to clean up 2 terms, drag and drop. I'm not going to save as a thesaurus because this took me 30 seconds, maybe. But for other cleanup where you take a little more time, it's easy enough to save that file and then you can recreate it later. So there's my single term for my indication. I'm going to select that. That just tells the script what I want to filter by. I'm going to select my new script. It should run through and create a new mesothelioma drug development phase table that I can export right back to BizInt Smart Charts. And as I do that, you might notice that there is also a normalized highest phase that you get for free along with this filtered table. If you're planning ahead, like I fortunately did this time and don't always, there's only one chart open, so it's easy to choose which one I want to apply my VPSCE export to. And that gives us 2 new columns. If I use view columns, I can find them and reunite them with the original data. I'm just going to drag and drop things a little closer together. And if I look at these 2 columns, I can see that this new column from VPSCE really is just a normalized all Arabic rather than Roman numerals version. So I'm going to go ahead and hide the original. Whereas over here we have the new filtered drug development phase table, and you can see again we didn't find a match here, and that's because this record was brought in by that Pipeline Plus search. If you were conducting the search in the original databases, you should certainly expect to find at least one match for your indication in each record there. And what we really want for our visualization is what is the phase for my indication? And the way to get that is to select that column, right-click, extract high phase in column. That's going to run off and give us one more column. Again, view columns, I will grab that, reunite it with the other phase. And so now we have a phase column that's just mesothelioma, which can be quite different from the actual overall high phase. So if we were to build visualizations with one versus the other, we would get a very different picture. I think we'll show one example of that. Right now, we're still in BizInt Smart Charts. We haven't gone to reference rows yet. We're about to. And we stay in BizInt Smart Charts for this kind of cleanup, extracting high phase, because we want to get that from all of the records before we go to reference rows. And then in reference rows, we can decide which value to use to represent the drugs. Okay, so we're about ready. I think I can do it. I'm going to save as at this point. You don't have to do that. I tend to have a before and after file, just in case I need to go back to the original chart. And that's simply because while I can hide these new VantagePoint columns, I can't delete them. Just like you can hide rows, you can hide other columns, you can't delete them. Okay. So now we can send to reference rows here or with the handy dandy icon. And we can tell that this sample chart that I started with, I had already identified common drug names. If I hadn't, this would not say regenerate, it would say generate, and I could not proceed because without common drug name, reference rows doesn't know which records to bring together, which records are about the same drug. So here I click next, and you can reorder databases in your preferred order. Maybe I'll move pharma projects up. And then I tend to click Finish simply because I can set any rules that I want to after the chart is created. So here is the reference rows view of that same set of data. Now we're numbering by group or by drug rather than by individual record. And I can set rules for any of these columns. And if I was producing a report, either in something like HTML or Word, I might set some additional rules. Here my goal is to prepare the data for building some visualizations. And so I'm only going to set rules for the columns that I need to use. I also want to make sure those columns are visible so that they'll be available to me. So the first thing I want to set is rules for these phase columns. So I can right-click, edit column rule. The default is just to choose by your database order that we set. Here I'm going to use this rule called most recently updated. So I want to take the phase, both overall, right-click again, edit column rule, and the indication phase. I want to take that from the record that's most recently updated, and we hope that that's also the most current phase. I'm going to do something a little bit different for the drug name, because I'm going to need a drug name for things like the piano chart, bullseye, the pharma visualizations that tend to plot one box for each drug. So I need a drug name, and I also would really like that drug name to be short. So we have this rule called least content characters. So that's going to select the shortest drug name. You can see there are a number of longer drug names, but in fact, in this case, this one is the shortest. So that's a way of getting to a shorter drug name. There are additional tools within VantagePoint to further clean those up. And I could continue setting rules, but I think those are the two things that I'm going to look at today. I also want to make sure I have something to use for a category. And this, you can see, is an ATIS field, but is what I used as a basis for the category. Those examples you saw in the presentation. So that's good that that's there. I think we will save here, save early, save often, and export again to VantagePoint. But before we do that, just to avoid confusion, I'm going to close out of this earlier VantagePoint. We don't need it anymore. We've already extracted highest phase, so we can close out of that. And now let's export. Again, we choose the last option. Again, it never hurts to give a somewhat descriptive name to that VPXI file, but it should automatically launch VantagePoint and import for you. Okay. So again, the columns have now become fields. You see we have the highest phase overall, and then we have this phase, which is the highest phase in mesothelioma. And we have this category. But you can see that, first of all, the coverage is only 75%. So if we're going to use this, we're going to need to do some further work. And when I tend to create categories like this, I tend to use all of the tools. I'll use BizInt Smart Charts for some review, because I can easily see across the record, as well as have the option to view the record. So I can get all of the information in one place. And then I also use the tools in VantagePoint for cleanup. That's definitely an important topic, and certainly something we could set up another webinar to talk about. But since we're primarily sort of showing some behind the scenes, how did we get to those visualizations you saw, we'll kind of work with a file I've created earlier for that. So just for demonstration purposes, to show that difference between overall high phase and indication high phase, I'm going to only visualize a subset of these chemical and biological classes. Everything with 7 records and above. I can select and add selection to a group, and kind of use sort of a placeholder name for this, because when I build the bubble chart, I can choose to only only use a group rather than the whole field, which would be quite a tall chart indeed. And before we create our visualizations, especially the bubble chart, we need to save. So let's go ahead and do that, just to be sure that I'm not overwriting anything. I'm going to save it with a new file name. Okay, the bubble chart is sort of at the top here in this list. And like many of our visualizations, these options windows will remember what we used last. So it remembers I want the rows to be the classes, and I want to use just that group. The one thing I am going to change is I want to plot first by that overall high phase, and then we can go back and look at mesothelioma phase. Some other options you can use. You can certainly give the chart a title, which I recommend for non-demonstration purposes. It's going to build a little matrix, and then it will build a bubble chart for us. You can also see that we're working with the full range of phases, and I think this will be an example of one more thing that I want to illustrate. Yes. Okay. For those of you playing along at home, you might notice that it is listing the full range of phases, but there is a little bit of a problem, and that is that launched is ahead of clinical, preclinical is after that. That probably is not what you want. So you want to make sure that your field that you use as phase is identified properly as data type phase. It isn't yet. We can right click, set data type. For version 10, which this is, phase is under custom, otherwise it will be listed here. Okay, let's try that again, once more with feeling. Okay, so now we have everything set. You can see behind me it is set as phase. And this uses a custom sort that should sort the phases in phase order as opposed to alphabetical order. Ah, isn't that nice? Preclinical is before clinical, which is before launch, and we can actually look at this chart. So small molecules and monoclonal antibodies are sort of the two top categories here, and you can see there's a cluster around phase 2, but there's also a big bump, especially in the small molecules at launch, with some clustering in the inactive phases as well. I'm going to run it one more time, and we'll just change one thing, which is, how about phase in indication? Okay, and we'll run through and build our chart. You can see the phase 2 looks, fairly similar. Drugs that are in phase 2 overall appear to also be in phase 2 for our indication. However, that giant bubble at launch has disappeared. So there actually are very few drugs that are launched, but a significant number that have been discontinued or current activity unknown for this indication. Okay, so now I'm going to switch over to one I've created earlier and sort of show you some of those visualizations being built. Okay. It's going to look very similar. It's the same set of data. It's just that I now have a highest phase mesothelioma. I've done some cleanup of the drug name. And I've also done some cleanup of that chemical biological class. You can see by the name of the field, there are multiple iterations of cleaning involved to get to a nice category set that we're using. So in that case, let's build a pipeline, piano pipeline chart. Again, it remembers my selections. I will probably use them. Drug name for the labels, phase and indication for phase, chosen to add a company name to the boxes, and we're going to color code by that category field that was created. I think for both this and the bullseye, we'll choose the PowerPoint export. So it runs through this list of 64 drugs fairly quickly in VantagePoint. Remember that in PowerPoint, these are each drawn as individual boxes. We'll probably see again some live example of why this set may not be the best fit for the piano, both because of the large number of drugs and because of the fact that rarely are the drugs evenly distributed amongst the phases with the same number in each phase. It will alert us when it's done. For those of us that like watching it run through, you can see that it is in fact drawing all the boxes and then formatting them as it goes along. There are 64 of these, so it will take a little bit of time. Again, that's probably the primary reason why we want to go ahead and switch to the bullseye once we get this demo built of the piano. Again. So yes, A, it's going to have to compact a large number of drugs into a single slide, and B, it's formatting a lot of individual boxes. You can see that when we added the hyperlinks, they're there. We could still go to each one of these individual drugs for review. We could go in and we could view the slide and almost make out those drug names, but this is probably not the best match for this data. If we were to build this for BizDash, we do have a way to break this into 2 columns. That probably is the best way if we wanted a piano chart presentation for this. Okay. Oh, I was impatient. I already saw my new chart, and now let's see what it looks like as a bullseye chart, because 64 drugs, that's probably more in a bullseye range, which I think I've created with up to around 100 drugs, and it looks fine. Of course, the more you have, the less space you have available for each drug to be plotted. Again, I am building this in VPSE, but we are definitely still working on it. So if you wanted to see what it looks like with your data, you would need to send us the BPT file, the chart file, so that we can work with it. Again, it remembers my field selections, and I am betting that these look good. Drug name looks good. Mesothelioma phase, and then that created categories. And certainly, again, were this not a demo, why not add a chart title? Because we're creating these in PowerPoint, it's probably just as easy to edit the chart title there. Okay. Again, walks through that list of 64 drugs. This should be a little bit faster because instead of having to draw one box for all 64, we're just plotting a point on a circle. There we have it. A small point. My computer is set up for widescreen. Should you have standard, that should be no trouble at all, because this is a circular presentation. You just might want to move the key a little bit over. Because it is generated in PowerPoint, as I say, we can feel free to customize as we wish. Let's see, I don't know, maybe can kind of make small adjustments. And this is that sequential color scheme that Diane mentioned. It was taken from Color Brewer, because not only do the colors move progressively, but the saturation of the color changes as well. It makes sense to change the font color for these very inner launched drugs. And you can change that for an individual point if you like. You can also format those data labels and place them. They are positioned initially for you, but you can do things to change that. For example, that might make more sense above. Let's see. I would like my screen to let me click that. Excellent. Okay. Now that GoToMeeting is out of the way, and we can continue to move things as we wish. You could do things again, like placing it above, or you can actually drag and drop to kind of nudge things down a little bit, and it will place a tooltip if it needs to do so. You can see that shorter drug names are certainly better. The longer they are, the more likely some repositioning of labels would be necessary for things to fit. The points for each drug will be placed evenly within the slice, but it doesn't have any particular meaning, just whether it is in a slice and which ring it's in. So these would be your phase 1 small molecule drugs, whereas the 2 launched are also small molecule. I think those are the 2 we wanted to see. Hopefully you sort of see how those were built. And I think I have one sample to try to build. I've built some of these for some other projects with different data, but probably more examples would be helpful as well.